So my presentation is, or my topic that was assigned was the epigenetics, the role of epigenetics in asthma development. This is,
Hannah, first tell people there are people I don't think who know who you
oh,
are.
I'm so sorry. Yes, hi, I'm Dr. Frudden, I'm Hannah. I'm one of the first year fellows, super excited to be here. I have no disclaimers because I am a first year fellow. So my topic was the role of epigenetics in asthma development. This is a pretty broad topic and one that I wasn't super familiar with. So instead of going into a lot of specific epigenetic findings and their related genes and phenotypes, I figured I'd do a quick little overview of a few things and then just talk about a few studies and their findings that I thought were particularly interesting, because this is a relatively, I'm hesitant to say newer, but there's a lot of, it's a area where there's a lot of new stuff going on that's pretty exciting. So there's a lot of different, and I'll kind of highlight places where you can find more information and just kind of how broad all of the findings going on in epigenetics research are. So asthma, as we all know, is a super complex disease. Both genetic predisposition and environmental exposures interact to cause the disease. These are just a few different things here because I wanted to highlight how, as you guys already know, but for this presentation, wanted to highlight a lot of all the different things that go into the development and morbidity and mortality involved in asthma. Just last week, Patricia went over the microbiome. As you can see in here, genetics, which is the study, how genes interact, and then epigenetics, which is what I'm going to talk about today. There's also just talking about like the environment, lung-brain axis. As we know, there's a lot of things that go into what affects and causes asthma.
Before we go any further, could somebody tell her how to take herself out of the image here? It's nice to look at you, but you're covering up part of the slide.
I wonder if I can, okay. I'm so sorry, you guys. I knew that this would be a struggle. I wonder if I can,
Is this just something video off?
yeah, let me try that. If I turn my video off, can you guys, am I blocking everything?
Not you, now just a big image of your name.
Mm. Hmm.
I mean, we can go ahead with, I don't want to slow you down.
I'm so sorry. I wonder if, oh, yeah, I don't know how to fix that. Um, so this is just a schematic showing the way that our environment can interact with our genotype, and that's what leads to the epigenotype. The term epigenetics actually means on top of conventional genetics, which I think is just kind of interesting. It refers to modifications that are a heritable regulation of gene expression that occur in the absence of changes to the DNA sequences. So there's a number of different molecular mechanisms that are involved in epigenetic regulation of gene expression. I'll go over a few of these in just a bit. But the really cool thing about, and then the epigenome as a term refers to the collection of epigenetic modifications across an individual's genome. So it's influenced, as you can see in this schematic, primarily by environmental exposures and genetic variation. One thing that's really cool about the epigenome or epigenetic changes is that unlike DNA sequence changes, epigenetic marks are dynamic and reversible. So they can actually serve as a therapeutic target, as well as biomarkers, as like in identifying molecular subtypes in heterogeneous diseases and other ways. So I'll highlight a few of those in some of the research studies I will bring up. This is just another schematic highlighting the differences between the genome or the DNA code and the epigenetic code. We don't really need to get into this. it kind of just is another way of showing some of what I just talked about. So these are the three main fundamental mechanisms of epigenetic gene regulation. I hope you can see the top of my screen, but number one, DNA methylation, which is what I'm going to primarily talk about during this presentation. It is the addition of a methyl group to the 5' position of cytosine in the context of CpG dinucleotides. And the second one is the histone code, or particularly acetylation of histone tails. This leads to post-translational modifications. Or the histone code post-translational modifications of the immunoterminal tails of the histone proteins can affect the packing of these and how they get transcribed. And then the third way is RNA-based mechanisms, which I still don't entirely understand that really goes into it, but that's talking about things like microRNAs and other non-coding RNAs that can affect downregulated effects. So the only one that I'm really going to touch on today is DNA methylation. This is because it is the most common epigenetic mechanism studied in asthma. More recently, there's a lot more interest in looking at the histone code and acetylation of histone tails in asthma research, but this is, historically speaking, an easier mechanism to study and where a lot of the bulk of research in asthma lies. So this is what I'm going to focus on today, again, just because epigenetics is such a broad topic and I kind of wanted to narrow it down for you guys. So as I just mentioned, DNA methylation refers to the bonding of a methyl or a CH3 group to the 5th carbon of a cytosine base to form methylcytosine or 5-MC. Depending on its location, on the location it occurs, DNA methylation can either negatively or positively correlate with levels of gene expression. So historically speaking, I think this shows here. So historically speaking, methylation was thought to depress or silence transcription, but as you'll see in a few of the studies here, research more recently has shown that it actually just kind of depends on where the methylation occurs and also can be even gene and cell specific. Typically, you will see, and this held true for the research studies that I looked into, methylation of a promoter region, which is what this schematic on the right side is showing, does tend to silence transcription or depress transcription, which makes sense when you think about what a promoter does. Just wanted to bring up CpG islands are just these short DNA sequences that are rich in CpG dinucleotides. They're usually found in the promoter regions of genes, which is why, again, going back to what I said, methylation frequently silences genes. But just of note, they can be found in exon introns and in other places. I just want to make sure I covered. And then also just going back to the CpG islands, about 60% of human genes have CpG islands. So that's why this is a really good area to study. So this is just another schematic showing how epigenetics also plays a big role in asthma, looking at how epigenetics and environment all interact together. This is from a study, and actually I think this is from a review article, which I think is, if this is something that you're interested in, this was a really great article for getting started. It's from 2023. This is a schematic showing, so GWAS refers to genome-wide association and EWAS refers to epigenome-wide association. The Venn diagram there, you'll see, just kind of shows how we're working, so how epigenetics is working synchronously with genetic studies to look at how the epigenetic modifications that we're looking at, and this is looking particularly at wide genome and epigenetic wide studies. But I just wanted to highlight here how, as we are finding out more on the epigenetic side of things, using that in conjunction with what we know on the genetic side of things, we're finding a lot of genes that are implicated in asthma. I won't then also just to highlight why I'm not going into more details. So just in there's several catalogs of specific genomes and epigenetic markers, and this particular Venn diagram is referring to the NHGRI-EBI catalog of genome-wide association studies. So kind of just highlighting it, there's 3,129 variants annotate to 1,576 genes based on the nearest gene. And then the NGDC Epigenome-wide Association Study Atlas contains 13,677 CPGs associated to asthma, which are annotated to 5,861 genes. So that's, I think, just to highlight why I didn't want to go into a ton of specifics and why this is a super interesting topic of study. And I think I got really excited actually reading about it, and it was something I hadn't really experienced before. So it's definitely worth looking into. And I think there's a lot of cool things going on in it. One thing that I just wanted to highlight, because it's something that I was unfamiliar with going into this presentation, and I think is very helpful in understanding these studies. So pathway enrichment analysis, which forgive me if you guys are all familiar with this, but I figured I'd put it in for anyone who wasn't. It is a way of summarizing a long list of genes into smaller lists of pathways with multiple associated genes. So basically, you take the differentially expressed genes in a study population, in your study population, to and you compare it to background genes to see if any of the pathways are overrepresented or are overrepresented. So as you can see here on the bottom, and this is used in a way, and I'll show you, there's a few figures later, but this is also used in epigenetics, where you basically map the epigenetic changes to, you can do it to genes too, but you compare your list of differentially expressed genes, either under or overrepresented, and then you compare them to the background genes. So for example, the entire human genome, and then you use a list of gene sets, which is a group of previously defined, based on other previous research, group of related genes. So like genes that we know from previous experience that are related to Th1 differentiation, for example. And then you see which of your genes, so in this example, STAT5, NFKB, IL2R, which let's pretend we didn't already know that. And then you compare that to, or I guess, yeah, so you would say these genes are overrepresented in our study population. We compare them to background genes, compare that to the list of gene sets that we already know. And then we can see that Th1 differentiation is overrepresented in our, or pathway is overrepresented in our study. Just wanted to get that out of the way because it'll help with everything moving forward.
I'm not sure I fully understand that. So go back.
So basically what
Differentially expressed genes in whatever disease population, is that what that means? That's the way you help find genes that you might think are involved in asthma?
Exactly. So say you have like a case and a control study, and your case study has asthma and your control study doesn't, and then you find that in your case study has overexpression of these genes highlighted in orange, and then you compare that to, so you use so basically you input all of this into like a computer system that does this for you, which I won't be able to explain why, because that's far above my pay grade. But it basically compares all of the genes that are overexpressed, for example, in your case population or your study population, compares that to a background set of genes. So we'll say the entire human genome in this example. And then it sees which list of genes, which is previously defined, are overrepresented in your study. So say say your study population, your asthma patients in your study population have an increased expression of STAT5 compared to the control. That looks at then the whole human genome and looking at knowing that Th1 differentiation includes STAT5. then it can basically jump to the conclusion that your study population has overrepresented pathways in Th1 differentiation. So patients with asthma in your study have Th1 differentiation pathways overrepresented. Does that make more sense?
Yeah, well, what does that tell you though about epigenetics?
So you'll see as we go forward, it basically, so in a lot of these studies, it maps these specific epigenetic modifications to specific genes and then further extrapolates that to overrepresented pathways. I think it'll make a little bit more sense when
All
we
right,
get
go
into
ahead.
this. So this is just another schematic showing that DNA methylation profiles using both candidate genes and epigenome-wide association studies in asthmatic populations are being currently used to elucidate biologic mechanisms that influence asthma susceptibility and severity. So either DNA methylation, it's just kind of showing like, is it a mediator? Is it unrelated to phenotype, or is it a consequence of the phenotype? In other words, can it be used as a predictive and diagnostic biomarker? Can it be used as a diagnostic marker only, et cetera? This will come up in our studies too. So here are some selected examples of consistent gene annotations for differentially methylated CpGs associated with environmental exposures that can be related to allergic disease and to asthma. I'm not going to go into all of these. A few of the studies that I will talk about highlight some of these. This is more just to show you how many different genes and epigenetic mechanisms are being investigated. Again, so these are some selected epigenome-wide association studies. I won't go into all of them, but this is just to highlight, again, some more genes that were mapped to these epigenome changes. Some of these probably look familiar to you. These are, again, more studies. So this is mostly, I put this in here if you are interested and want to learn more about this. I read through a lot of studies. I highlighted the ones that I thought were cool and also the ones that I felt were easier to summarize and had more important findings, but there's a lot of really cool research out there. I read a lot of studies for this presentation and I actually really enjoyed it. So one of the biggest ways that epigenetics are being used in the study of asthma are looking at DNA methylation in the developmental origin of asthma, so alterations in the epigenome in the intrauterine environment, leading to postnatal disease risk. A big risk factor that has been studied is in utero exposure to maternal smoking. This study was a really cool study. This is an epigenome-wide analysis of DNA methylation. It looked at both the fetal lung and the corresponding placental tissue to see associated CpG sites that were differentially methylated and then mapped those to different genes. So the most significant CpG sites mapped to, and I'm trying to remember, is this? Okay, perfect, yes. So you can see here, so this is what I'm talking about when I kind of talk about enrichment or pathway enhancement. So the name on the side here, those CG, those are the DNA methylation associations, and then you'll see they map to specific genes. So that's where the pathway comes in. So this study was really cool. So you'll see here the on the left side is the fetal lung analysis. So the most significant CpG sites in the fetal lung analysis mapped to, and I've highlighted them in red, but the PKP3, ANKRD, 33B, CNTD2, and the DPP10 genes. And then on the right is the placental methylome. And this figure is just showing the most significant CPG sites mapped to and the genes are up there at the top, but the GTF2H2C and the GTF2H2D genes. What was really cool is that there was 101 unique CPG sites with that were statistically significantly concordant between lung and placental tissue. And then gene set enrichment analysis, so this is what I was talking about before, demonstrated enrichment of specific disorders such as asthma and immune disorders. So this is
Go back a second. I'm not sure I understand that. Go back. So is this plot on the right
this one.
showing that those five, no, go back to that one. These 5 bars down on the bottom here,
Yeah.
which show it keep switching. Those are the significant ones, those particular 5 sites where methylation occurred were of significance because of nicotine exposure.
Yeah, so you can see, okay, so the red bars were intrauterine exposure to smoke, and the blue bars were not exposed to smoke. So then down at the bottom are all the DNA methylations, you can see where it says CG. So the highlighted in pink are ones that have a statistically significant difference in methylation. And you can kind of see that, you can see that represented in the box plots here, where you can see that the orange bar, which is the smoke exposed, has more methylation compared to the blue bars, which is the, oh wait, sorry. Yes.
Yeah, I think that's right.
Yes, compared to the blue bars, which has no smoke methylation or no smoke exposure. So those 1, 2, 3, 4, 5 CPG sites map to the genes up at the top, the GTF2HC and the GTF2H2D.
And what do we know about the function of those genes?
That's a great question. I don't actually know off the top of my head. So those, for example, could be great genes to look further into with regards to asthma. And I apologize if somebody does know what those genes do. It did not mention in the paper. So anyone feel free to speak up if there is something that we know that those do.
I don't. All right, move along.
So this is another study. So this was a meta-analysis. This is again looking at maternal smoking. So this looked at 3 different cohorts, or actually, no, sorry, 13 different cohorts with a total N of nearly 7,000. It was 6,685. And it was looking at maternal smoking in pregnancy and newborn blood DNA methylation. It looked at over 450,000 CPG sites. So of the total N, 897 were exposed to smoking and 1,600 were exposed to sustained maternal smoking, and 1,646 were exposed to any maternal smoking during pregnancy. So over 6,000 CPGs showed differential methylation at genome-wide significance. So if you look to the left here, so this is a Manhattan plot. These are something that you should definitely know how to look at if you are interested in epigenetics, but they're really cool. And once you know how to look at them, they're really easy to understand. So basically, what this is showing is, so the x-axis shows haplotypes for each region of the gene that was tested, organized by chromosome. So each chromosome has an associated block. So you can see there's the X chromosome is not represented on here, so that's why there's 22 of them, but you can see like how they alternate between dark gray and black, or I guess light gray and black. And you can't really see from far away, or I guess you can kind of infer that each block, it's not a solid line, it's composed of thousands of dots, and each dot is one haplotype. So as you see, like, as the dots go higher and the little kind of, I guess, block, I'll call it that, goes higher, the more, basically the more representative. So I'm just trying to, so the y-axis is the probability that there was an association observed. So, and I'm sorry, I just want to make sure I'm saying this right. So the higher it is, the higher the dots rise and the higher the block rise, the more likely there is to be a increased occurrence. That probably didn't make very much sense. So sorry, so starting over. So with regards to the y-axis, it represents the relative frequency or the association of a haplotype in that region in affected individuals compared to control individuals. So the higher the peak, the stronger association with the trait, if that makes sense. They're kind of hard to explain, but basically, so like, let's look at, on this one, you'll see like chromosome 1, chromosome 7, the higher up it rises, the stronger the association with the trait, in looking at your comparison between study populations. So on your right, this is a volcano plot, which is used for a lot of different things. In epigenetics, a lot of times, what volcano plots are used to show is the direction of effects for methylation. So on your left side would actually be hypomethylation, and on your right side would be hypermethylation. So in this study, which I actually, going back to kind of what I had said earlier about does methylation cause repression or not, in this study, they actually were kind of equally sparse between hypo and hypermethylation. So this is the same study, and this is just showing, again, there were a lot of different CPGs that were differentially methylated at a genome-wide significance. The top hit in all newborn models was a DNA methylation CG05575921, which mapped to the AHRR gene, which has been observed as to be differentially related or differentially methylated in relation to smoking in many studies of adults and children. They did enrichment in pathways on this data set as well, which showed it mapped to processes critical to development. So this is kind of just going over again what I already said. They were, I think top finding what I talked about, kind of just highlighting that there were a lot of different CPGs, fewer statistically significant findings for any smoking compared to sustained smoking, which I think makes sense from a logical standpoint. And then they did, so in 5 cohorts, there was data for the same children at an older age, I believe 5 years of age, and it showed that the effects persisted. So knowing all of this, one really cool study, actually a couple of
I'm going to interrupt you again. Go back.
yeah.
This is not easy stuff.
No.
Not that one. The summary one. The summary one. The next one. This one. No, just stay on that summary one. Not that one, the one with text. That one. So the bottom line, if I understand this correctly, is one particular site was the most statistically significant one altered when mothers smoked, and there was DNA methylation statistically significant at that site, and it persisted into life
So,
after birth. Is that the bottom line?
so this study, yes. So this wasn't necessarily looking at asthma specifically. This was just looking at DNA and methylation in cord blood of newborns and their relation to smoking. So this study didn't even incorporate asthma. What it was looking at, so it looked at, so the CPG that matched to this gene AHRR was the most statistically significantly associated with maternal smoking exposure.
Yeah, I understand it. It didn't relate to asthma, but it did show the effect of maternal smoking at a specific epigenetic site in the
Yeah,
genome.
yes.
All right.
And when it talks about like the persistent effects, so there were others that were significant, so it looked at those as well, but the AHRR was just the most statistically significant, if that makes sense. Okay, so this the study that I'm going to talk about next is, I thought, a really cool study. It's looking at how can we use like looking at DNA methylation in conjunction with in utero preventative strategies. In this study, it's specifically related to maternal smoking. So this was an epigenome-wide study looking at DNA methylation in placentas from from mothers who smoked, and looking at the effects of or the impact of vitamin C supplementation on placental DNA methylation changes related to maternal smoking. So they looked at, so they took women who smoked and they supplemented them either with vitamin C or placebo. And then they also compared that to women who had, so they compared those two, and then they also compared women who had placebo supplementation to women who had never smoked. So this is, again, another one of those Manhattan plots. And I think this image probably does a better job than the first one. So you can see here that so the first one is all the women smoked, and then they were either supplemented with vitamin C or a placebo. And there was one significant CPG, which you can see here as the CG207-90161, which is located in an intergenic region of CD28 gene. So you can see here that kind of outline dot. So that's where I was kind of trying to explain that the higher up on the y-axis, the higher the association, or the bigger the difference between the two groups. Down at the bottom, similarly, there were two, or it looks like there were three CPG, three significant CPG sites identified. So this is where they did. So they did this I kind of didn't 100% understand, but basically what they did is they looked at, so this Venn diagram is showing like, okay, the candidate CPG sites between that were significant in the vitamin C versus the placebo versus the never smoker. And then they found the overlap of these 9,541. And that's what they used for downstream analysis. So in B, so this is a heat map. So heat maps are another thing that can often come up in epigenetics. And so what a heat map is, it's basically a way of showing, so you have so many genes across so many samples. So heat maps are a good way to help visualize, again, patterns. So if you can see here, like where these little guys are, usually that shows what kind of it is mapping to. So typically genes and then to pathways. What you really only need to understand for this is that if it's blue, it's undermethylated. The more undermethylated it is, and then that's just kind of chunked into pathways. And if it's yellow, it's more upregulated. This is this is basically showing, so those 9,541 CPGs they used to do downstream analysis, which is basically what this is. They looked at how these differentially these DMRs and canaves, how they mapped to different phenotypes. So they looked at, so you can see like let's say, for example, in the green, those are genes, those are pathways that we know are associated with wheeze. And then you'll see that those overlapped with a lot of things, with a lot of genes that were mapped to in this study. I know this is kind of a lot to look at. I'm probably not doing the best job of explaining it. But another way of saying this, so let's look at wheeze again. So within this study, they identified candidate CPGs that were associated with infant lung function. So 34 of them, that's the red, the lung development. Within the wheeze, there was one CPG at 12 months of age, and then they also found that increased methylation in DIP2C, APOH, and PRKCA associated with improved lung function and decreased wheeze in offspring of the vitamin C treated smokers. I know that's a lot of information. So I'm just going to summarize, showing that vitamin C supplementation in the pregnant smokers compared to placebo did decrease the impact of maternal smoking on offspring pulmonary function and incidence wheeze. And then based off of their downstream analysis, they reported that improvements in lung function were mediated by the resolution of DNA methylation changes identified in the placenta of the treated individuals. And then through that pathway enrichment that I talked about earlier, the reduction in DNA methylation in the placenta occurred at loci in pathways associated with oxidative stress, fetal development, and lung function. I know that's a lot and it probably doesn't make a ton of sense, but I do have a lot more to get through. So unless I'm not, I might just keep jumping ahead and it might continue to make more sense, hopefully, as I go on. But if there's any confusion now, let me know.
So just as a summary, so the point of vitamin C is that it reverses the methylation caused by smoking.
Yes.
All right.
So then they also looked at environmental toxins like pollution. So this study was looking at polycyclic aromatic hydrocarbons. It was looking at umbilical cord white blood cell. The cohort was 20. So this was actually, so they found over 30 DNA sequences whose methylation status was dependent on the level of maternal PAH exposure. And what this study is showing, or what this image is showing, which I just thought was kind of cool, it's basically using, basically like looking at DNA methylation they were able to distinguish children born to mothers with high airborne PAH exposure from those who were not, with the area under the curve being able to tell if they had been exposed. So what this is showing is that DNA methylation can be used as a biomarker for exposure to these environmental toxins. This is just talking about those specific methylation associated genes. So 6 sequences were found to be homologous to known genes. One of the biggest ones was methylation of this in gene ACSL3, positively and significantly associated with asthma classification.
This second statement here, methylation of whatever that is, ACSL359CG, was positively and significantly associated with asthma classification, seems to suggest a very strong outcome that polycyclic, or basically air pollution, significantly altered one site, and that one site was highly associated with development of asthma.
Yes.
Did I read that correctly?
Yes, that is, you are reading it correctly. A different study that I'm going to talk about just to because now we're getting into genes that I was more familiar with. So we're all familiar with IFN gamma and IL-4 in the pathophysiology of allergic asthma. So this study looked specifically at methylation changes in IFN gamma and IL-4 genes in the context of diesel exhaust particles. So looking at, so in vitro studies have shown that demethylation of sites at the proximal promoter and conserved intronic regulatory element, or CIRE, in the first intron of the IL-4 gene, and then hypermethylation of sites in the IFN gamma promoter result in efficient IL-4 production. So going back to this, we know that IL-4 leads to naive T cells to differentiate towards Th2 cells, leads to B cells class switching and producing Ig. So in other words, leading towards allergic asthma. So then also in vitro studies, Th1 differentiation associated with methylation of highly conserved DNase I hypersensitive region at the 3' end of the IL-4 gene. So those are in vitro studies. That's what this is showing. This was a murine model looking at DEP and allergen exposure as it alters methylation of T helper genes and IgE production in vivo. So they took these mice, they exposed them to DEP for 3 weeks while also undergoing intranasal sensitization to A. fumigatus. And then they looked at total IgE, which increased following sensitization to aspergillus, and then this was further augmented when combined with both A. fumigatus and DEP. This figure is just showing IgE production following exposure to both of these. You can see the highest IgE was observed after dual exposure. And then they looked at methylation of the IFN gamma and IL-4 gene following these exposures. I'll just kind of summarize for you. Basically, demethylation of sites at the proximal promoter of the IL-4 gene led to increased IL-4 production, Th2 cytokine production. I put that in parentheses because that's just kind of how we know it works. It wasn't actually shown in the study. And then hypermethylation of sites at the IFN gamma promoter also led to IL-4 production, which also makes sense. So this is looking at specifically, so this next study I'm going to talk about, again, just to This is looking at DNA methylation of FOXP3 and IL-10, which are, this is the schematic just to remind you of how this can have a play in asthma. FOXP3 is responsible for the expression of Th2 following exposure to allergens. It's required for the immunosuppressive function of Treg cells. And we know that based off of previous studies, FOXP3 gene is reduced in patients with asthma and allergies compared to healthy control. And then expression of IL-10 can help induce and maintain tolerance to allergens aerosol, and it's known to inhibit dendritic cell maturation, eosinophil survivor, and IL-4-induced IgE synthesis. In asthmatic patients, previous research has shown that IL-10 production is also diminished. So this is a study that was looking at methylation of FOXP3 and IL-10. I know this is kind of a busy study, but to summarize, basically what it showed is that there were differentially methylated regions in the FOX3 promoter and IL-10 regions that were preferentially methylated in asthmatic patients. So in patients with asthma, there was a significant association with methylation at 4 CpG sites in the promoter region of FOXP3. And asthma was significantly associated with methylation at all 3 CpG sites in region 4, intron 4 of IL-10. Then they looked at methylation and air pollution. So exposure to air pollution was associated with increased DMRs in the promoter region of FOXP3. And then they did a, so the way that they did the study was kind of confusing, but at the 90-day time period, so basically they were able to measure the ambient air pollution levels. They had basically, it was kind of a cool study. They like had a weather, like a weather place set up, and they would measure this. But they found that increased exposure, they looked at 4 different ambient air pollution, so NO2, CO, and PM2.5, and then one other one that I kind of forget, and O3. So increased NO2, CO, and PM2.5 90 days prior to when they measured the blood was positively associated with FOXP3 methylation. They did not find any significant associations between ambient air pollution levels and IL-10 methylation, though. So since we're running low on time, I'm not even going to get into this, but this is the same study. So in other words, basically looking at activated Treg, so CD25 high, CD127 low, and CD4 positive cells and protein expression and methylation. They did that in a subset of patients. They performed multivariate linear regression, and they found that Tregs were inversely proportional to the average FOXP3 methylation of the CpG sites in the promoter site. Didn't find any significant association between FOX3P methylation and FOXP3 expression, and then IL-10 cytokine expression was positively associated with IL-10 methylation at region of intron 4. But I think really the important part about this are these studies here showing that there is an association between methylation and asthma status and similar methylation profiles when you look at ambient air pollution. So thinking about how air pollutants can alter regional DNA methylation, how that is also associated with asthma was the whole point of this study. Since I'm so sorry, I think we only have like 10 minutes left or less, I'm gonna just kind of try to keep pushing through. I think I've got 2 more studies. So this was looking at stress as an exposure to DNA methylation. So this is something I thought was really cool. So they looked at exposure to violence and chronic stress in children with and without atopic asthma. So these ETV, EGTV, CDC, they all refer to different types of measurements of stress and violence. So ETV is exposure to violence scale, ETGV is exposure to gun violence, CCDS is checklist of child distress symptoms, and then PSSS is maternal perceived stress syndromes. And then they looked at CpG sites on all of these. So what this study, so what this particular figure is showing is that association between DNA methylation and these measures of stress and violence. So there was some significant sites of methylation that were found to be more overexpressed or overrepresented in different measures of violence. So in the PSSS and the maternal perceived stress syndrome, you'll see methylation of CG blah, 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 which is in SNN on chromosome 16, and this other CPG site in PTPRN2 on chromosome 7 were associated with maternal perceived stress. And then in the gun violence methylation of this CPG site in LINC 01164 on chromosome 10 was significantly associated. In this study, and I think this is, yeah, the same. So in this study, this is the same. So in this study, unfortunately, they didn't find all of these, none of these specific CPG sites were associated with atopic asthma in the original cohort. But some of these other CPG sites, they compared them to, so in the original cohort, which was a group of Puerto Rican children, then they took 2 other cohorts, so they combined them for all a total of 3 cohorts, which is what this figure is showing. This is looking at all of these different cohorts, EVPAR, Project Viva, and PIAMA, which I forget what that stands for. So in this one, they did find a couple of genes, which I've highlighted here in red, and their associated DNA methylation CPG sites. PLCB1 was associated, and it's associated with bronchodilator response in children with asthma, and is known to be differentially expressed in children with treatment-resistant asthma compared with children with controlled persistent asthma or healthy controls. And then in other previous studies, another gene, TMEE 196, has been associated with decreased odds of NSAID exacerbated respiratory disease in asthma. So this study, in other words, didn't show necessarily that within the original cohort that these were specifically rated, but these CPGI sites were all also differentially methylated in violence across these cohorts, as well as asthma. And on the side there, that's me talking about some other studies. So this is my last study, I believe. So this is just looking at DNA methylation as a predictor of later in life asthma. This was looking at one specific gene, HK1, and expression in cord blood being predictive of adolescent asthma. I think this is still the same study. So basically, what they did is this was an epigenome-wide screening, and they identified 10 CPG sites associated with adolescent asthma, 5 and higher expression of HK1 in cord blood was predictive of wheezing in infancy. So basically what this is showing is they identified some novel associations between asthma and wheeze, specifically at this one CPG site, CG 166589191, and the expression of HK1. So this is kind of just using, like, can we use cord blood as a predictor of asthma development later in life? This is a similar, this is actually a different study, so I guess I had 2 more. So this is a different study. This is looking at differentially methylated. It's a meta-analysis looking at differentially methylated regions in cord blood DNA associated with childhood lung function, asthma, and chronic obstructive pulmonary disease across the life course. So it was a meta-analysis. So you'll see here that there were several molecular locations of DMRs that were associated with different components of lung function, associated with different genes. And you can see here they were kind of over or underexpressed in children versus adults. All that is to say that DNA methylation can be a really good use as can be used to predict later in life asthma. So I did my best to highlight a few studies. There's so much more going on. These are all just studies, more studies. This is a great review that was done pretty recently if you're excited about this stuff. And then this is the word of the day. If anybody has any questions, I can do my best to answer them.
The word of the day is epigenetic.
I think it's epigenetics.
Yeah. So let me ask you, this is a very complex topic, obviously, but thinking about the interplay of genetics and epigenetics, do you have to be born, so to speak, with the wrong genes for epigenetics to make any difference? In other words, if you're born someone not to be atopic and not to be asthmatic, does it make any difference what exposures you have that leads to epigenetic changes? You'll never become atopic or asthmatic. Or does epigenetics a strong enough influence that it could, change that?
That's a really great question, and I think that's what a lot of really smart researchers are trying to figure out right now. What we do know is that the interplay between genetics and environment and all of that leading to epigenetics can really greatly alter what phenotype ends up coming out. So I guess my answer to that is based off of what I know about epigenetics, which is limited. I think we don't 100% know. Obviously, you need the gene there to be modified. But I think we just don't know enough yet to say for sure.
Yeah, I have no idea, but my guess is it's the answer to that's probably unknown.
What just to chime in, I think again, I am no expert in epigenetics, but I think the thought is that you may not need to have that genetic predisposition in terms of a specific change of the genes for this to really have an effect. And so the environment, as Hannah was saying, can cause these changes to folks that don't necessarily have genetic susceptibility. I guess the phenotypic impact of that in part depends on how far you tip them over, right? If the effects of the epigenetic changes are on top of overlying genetic predisposition and genetic risk, then I think certainly that may be a more powerful manifestation of whatever it is you're looking at. But I think the thought is even with in vivo studies in mouse models in mice where everyone is genetically identical, you can see changes depending on environmental stimuli. So I think certainly there can be interplay, but perhaps in certain situations, this can cause an effect regardless of genetic makeup.
So you're saying epigenetic changes are potentially strong enough to make a non-asthmatic asthmatic, or these are potent changes to the DNA?
Yeah, I think they can be potent, whether or not it's going to completely push someone into a disease state versus not. I think it's unclear into full-on asthma, but I think they are potent enough to potentially cause changes. And I think the other layer on top of that is how sustained are the changes, which certainly can be in part the degree of the exposure itself.
Yeah, I wonder about that. Once you have these, say, methylation changes, do you live with them that way for the rest of your life?
From what oh, go ahead.
So that's the thing about epigenetics, is they're modifiable. I mean, even if you look back at the first study second study with vitamin C, so they are changeable. So unlike the genetic code, the epigenetic code it's mutable. I guess where's this?
Any other questions? I'll just make a comment. So next week, I think is virtual only, but I think in 2 weeks we have a visiting speaker. So I'd like to encourage people to come in person once again, starting in 2 weeks. Where's Vinod? I can always count on him to ask a question.
I'm here, Len. Hello, good morning. It is a fascinating topic, and it is so complex. In terms of you did a really good job in trying to pull those things together. So I if I did have a question earlier, was the vitamin C and the effect on reversing. I was just curious in terms of how is that in terms of is there a dose and what's the mechanism, if at all, or is in terms of how it can affect. Thank you. Thanks, Len.
Okay. I didn't need to force you to create a question.
No, it crossed my mind. I didn't want to to stop the presentation at that time. So but it is curious in vitamin C. Vitamin C. We hear about vitamin C and all the other things that it does, it can or doesn't do, and over at least expectation of what it can do. But it is interesting to see vitamin C having an effect here. So I didn't know if you had an idea why and if what dose, what mechanism there is in terms of affecting epigenetics.
All right, thank you. Anybody else? So we're a little past 8 o'clock.